Thank you very much, Hazel, and thank you all 57 folks who I see are participants on this session at Land's Edge. Once again, I think this has been a great Science Days thus far, and I expect that this session is going to be just fantastic too. We've got some great talks. So, by the way, I am Steve Radkin. I am the coastal ecologist and limnologist at Olympic National Park. I've been here for going on 26 years now, and it has been a great privilege to be able to work in the marine environment here. The theme for this session is At Land's Edge, and the North Coast and Cascades Network is a constellation of parks that have a lot of really, well, not really unique, because you can't be really unique. They just have unique marine resources. They're archetypal marine resources. And even the parks that are not marine parks are heavily marine influenced. And these parks span from inside the Salish Sea to the outer coast on the Olympic coast and down to the northern Oregon coast. Today's talks cover the wide gambit from charismatic megafauna to charismatic microfauna and plants. And also what's represented in our national park units here in the North Coast and Cascades Network is a gradient of human impacts, also from global impacts in wilderness areas to parks that are experiencing more direct human impacts. And all these talks touch on aspects of those. So without further ado, we'll get into the talks. I'll just say for presenters, these are 5-minute lightning talks. And after 4 minutes, I will hold up a sign that will say 1 minute. And at the tail end of it, after your 5 minutes, I'll hold up another sign and I'll turn on my video at the same time during those things. Presenters, please try to keep your video on if you're comfortable with that, because it's nice for people to be able to see that human interaction. Okay, so without further ado, we'll get into our first talk, which is by Kimberly Nielsen from the Oceans Initiative. And she is going to be talking to us about behavioral changes in the endangered southern resident killer whales. So charismatic megafauna. Please take it away. All right, I'm not seeing you up here yet. I see your first slide, but I don't see it in presentation mode. And I believe you're also on mute. Okay, you're off mute now, Kimberly.
There seems to be a little bit of a lag. Let's see. There we go. Okay. Hi, everyone. My name is Kimberly, and I am a research associate at the nonprofit Oceans Initiative. And on behalf of my colleagues, I am excited to share just a little bit about our work on vessels and killer whales. Southern resident killer whales are obviously an iconic species. They're culturally and ecologically significant. This population, unfortunately, has been in decline for decades, and so they are listed as critically endangered. And as of January of this year, there are only 73 individuals left. And that's because they face multiple individual and compounding threats. So limited prey, mainly of Chinook salmon, contaminants, and noise. Our team has been monitoring southern resident killer whale behavior and habitat use in the San Juan Islands for over 20 years. I hope this works, but I have a sound clip from a hydrophone that we deployed off the west side of San Juan Island. And in this clip, we can hear kind of ideal natural conditions. So the whales are communicating with each other and echolocating to find prey. In reality, though, the soundscape in parts of the Salish Sea and certainly in all of Puget Sound is dominated by vessel noise. Some of my colleagues' work from the early 2000s showed that vessel presence has a negative effect on killer whale behavior. And in response to that work and to help mitigate vessel effects on whales, The state has instituted different regulations over the years for how vessels should operate around southern residents in particular. These rules are different whether you're a private boater or a commercial whale watch operator. And in the summer of 2021, WDFW initiated a commercial whale watch licensing program, which puts stricter regulations in place for them. So not only are there distance and speed limits, but there's also a limit on how many commercial whale watch vessels can observe the whales at a given time, and they can only be viewed in close proximity during two-hour windows. These new 2021 regulations afforded us a unique opportunity to look at patterns in whale behavior both before and after these new regulations. And so we observed both vessel and whale behavior from 2018 to 2023. We recorded all observed whale behaviors, but for the purpose of this study, we focus on feeding for a couple of different reasons. And that's because feeding is clearly important for survival, but also because the waters around the San Juan Islands are part of their summer critical foraging habitat. So these are historically very important feeding areas. Our team tracked whale and vessel movements from the west side of San Juan Island using a fully land-based, completely non-invasive setup. We have 3 sites along the coast of San Juan Island, and our southernmost site is an American camp, which is part of the National Historical Park. And that allows us an expansive view of both the Strait of Juan de Fuca and Haro Strait. Part of what we found was that there was no significant difference in the amount of time the whale spent foraging before and after the licensing program. The vast majority of recorded behaviors were traveling, which is shown in blue, as well as foraging in green. In recent years, we recorded very little resting and socializing behavior at all, and that's actually something that we're looking into separately. We also found that changing vessel regulations had sort of a mixed or nuanced effect on southern residents. On the left here shows that as the number of boats within 1 kilometer of the whales increases, they are much less likely to engage in foraging behavior. However, the box plots on the right show that vessel distances from killer whales have increased post-regulation, which is a good thing. So most boaters on average are giving them a bit more of a buffer. Overall, commercial whale watch boats are serving as both magnets and sentinels. So their presence does attract more private boaters onto the scene, but they model good behavior overall. So private boaters are more likely to stay further away from the whales and to reduce their speed. So overall, management must be adaptive. Commercial vessels often only represent a small fraction of the boats on scene around these whales. And even though these updated regulations seem to be diminishing the effect of boats on time spent foraging, at least at this site, this is just one little piece in a very complicated puzzle of killer whale survival, southern resident survival in particular. If you'd like to read more about all of those stressors and how they play into southern resident decline, we published a paper on this recently. This is a massive warning for the future of the population, but there is still room for hope. So vessel management is good progress, but we still have a lot to do to protect the population. So I just want to say thank you to the Science Days team, to San Juan Island National Historical Park for allowing us access to the site, to WDFW for funding this particular study, and to everyone here for listening.
Fantastic. Wonderful talk, Kimberly. Thank you very much. Once again, questions, please, if you have questions for any of the presenters, put them in the Q&A section. And at the end of the session, we will en masse address them, and all of the presenters will be here for that. So moving on, our next talk is by the estimable Amelia Ritger from University of California, Santa Barbara. And she is moving from charismatic megafauna to charismatic millifauna, razor clams, something that's near and dear to my heart, and impacts associated potentially with ocean acidification. So without further ado, Amelia, it is your turn.
Are we in presenter view right now, or is it just my slides?
You're in presenter view.
Okay, that's what I thought. Sorry. There we go. Okay, great. Thanks, everyone. Okay, so yeah, Steve kind of stole my thunder. I was gonna say if you are looking at a sandy beach ecosystem, these are the charismatic megafauna of organisms that live in the sand on the coast of Washington and Oregon. So, oops. Oh gosh, we're skipping ahead. So first off, Pacific razor clams are an important species found in sandy beach organisms. Their population densities vary between beaches across Washington and Oregon State, and also across time as well. But overall, in general, populations are more dense on Washington State beaches than Oregon State beaches. This translates into a dollar value. So the razor clam fishery actually stimulates coastal economies. For Washington State, that corresponds to around $40 million in income and $12 million for Oregon. The razor clam fishery is a significant source of revenue for coastal communities in Washington and Oregon State, and there are both commercial and recreational digs that folks can get involved in. But speaking of recreational digs, recreational digging is a significant cultural activity if you live in the Pacific Northwest of the United States. But I wanted to also highlight how razor clams are culturally significant to coastal tribal communities. And so here we have a quote from a tribal member from the coastal treaty tribes in Washington State, who says that it's beyond a recreational activity. We've been coming to the same beach generation after generation. I even used the same shovel my grandfather used to dig clams back in the 50s. Clamming holds a cultural aspect tied to the tribes who have been around long before. So there is this importance of this species to not only ecosystems, economies, but also cultural activities and cultural practices. But razor clams actually aren't just isolated to Washington and Oregon. They're found all the way along the coast from California up through British Columbia and even into Alaska. And this really broad geographic range corresponds to differences in environmental conditions that razor clams are experiencing across their range. And so, for example, along our coastline, we have this latitudinal gradient in temperature where further north you go, the colder the water gets, the further south you go, the warmer the water gets. But that nice linear latitudinal gradient doesn't exist with environmental conditions like upwelling, which actually exhibit more of a mosaic pattern. So you have this seasonal variability in upwelling, which can create more acidic conditions in some regions than others. And the idea behind this study was to understand how these different environmental conditions for these different razor clam populations across their geographic range might be influencing things like their shells. So what we did is we collected razor clams from quite a few locations along the coast of Washington, Oregon, and even up into British Columbia, Canada. So here we have sites in Long Beach on Vancouver Island. We have Claylock, Copalis, and Ocean Park in Washington State. And notably, Claylock is within Olympic National Park. We have Astoria, Newport, Coos Bay, and Gold Beach in Oregon. And so looking at things like shell thickness, we found that shell thickness actually didn't follow this latitudinal gradient where shells were thicker up north or thicker down south. There's actually this variation between locations where places like Coos Bay, for example, down in Oregon, have a similar length to thickness ratio as shells all the way up in Canada and British Columbia and Long Beach. And when we were crushing the shells to look at how much force is required to crush the shells, we found that there also wasn't a latitudinal gradient in between locations. And so generally speaking, the bigger the shell, the thicker the shell, and therefore the more force is required to crush that shell. But we didn't find this nice, clean latitudinal gradient. We found a lot of variation between locations in terms of shell strength. What could be causing these variations? Well, one, it could be due to that upwelling mosaic that I mentioned before. And when upwelling happens, it brings cold, acidic waters up into the shallows, and that causes acidic conditions, which could impact the shells, and that might be causing these variations. We also might be seeing regional differences in temperature, aragonite saturation, and phytoplankton that could be causing these observed mosaic of thickness and strength. But I also wanted to highlight that shell burrowing polychaetes may also be contributing to differences in shell thickness and strength, because when those polychaetes burrow into the shell, the clam actually thickens its shell in response, and it could be contributing to some of these patterns, such as what we were seeing up in Canada. None of this work could have been done without the help of many, many people and many different agencies. And so I just wanted to take some time to thank everyone who helped collect clams.
Fantastic. What a wonderful talk, Amelia. Well done. Really appreciate your efforts. Moving on, once again, if you have questions about this in the Q&A. Moving on now, we are going to go to Scott Smith from the Blue Shirt Justice League, Inc. And Scott is going to be talking to us about eelgrass beds and decline in eelgrass and potential relationships with environmental pollution up in the San Juan Islands, particularly in Garrison Bay, around San Juan Island National Historical Park, another place near and dear to my heart. Okay, Scott, please take it away.
Well, first of all, I want to explain some background and thanks to this team. This is really why eelgrass is dying in the eelgrass and seagrass are dying in the San Juan Islands is a major problem that needs to be resolved. And this is an example from people from all walks of life and multi-generational diversity coming together. Dr. Sandy Wiley Echeverry from Friday Harbor University of Washington Labs reached out to me along with Dr. Gary Green and student Avantika Dutt, who also has a poster session afterwards, were indispensable to this. So what we deployed is I have a technology that I use that is patented that I use for the nonprofit work and scientific work. And what it is using an open cell foam in the whole water column. And you see what's exposed over time. What we do is we deployed our initial deployment and had exposure over time because to date, no one's been able to figure out what has been causing the decline in the seagrass. And we specifically did the initial deployment in Reef Bay, and there's a map, off Shaw Island, and you can see, the ferry traffic, and I'll be talking about that in a little bit. This is the deployment in the water column, and I have a video that kind of shows it on the left. And you can see how we're deploying this in the water column, and these initial testing was out 21 hours and then exposure for 45 hours. And this is how we pull it. And this is made of unique medical grade material that is actually the same formulation as approved by the FDA for a medical device for humans. And you can see it's repelling the water and it concentrates contaminants. And the water column, this is our preliminary data. We identified diesel range organics, not gasoline. And this was the absorption or what accumulated over time. And our preliminary hypothesis is with post-COVID and the additional boat traffic and ferry traffic, just by definition, diesel can leak from all the boat traffic. And we identified that in the water column. And we also looked at metals. And we were able to rule out a lot of things, but we also, the hypothesis is it potential exposure to diesel range organics and possibly excess phosphorus, from landscaping and, as nutrients for potential harmful algal blooms or some kind of bacteria and manganese. So we have identified these and what this shows, top, middle, bottom, and what's going on in the sediment, what's been deposited. And potential impact sources. This is what we're out there. And you can see the ferry traffic. And we just got this data in October of 2024. 366 individual ferries passed by Reef Bay Net from Lopez Island Terminal to Friday Harbor. And again, this is, you just can't have diesel boats, gas boats. You're going to get some leakage in what is the traffic. The bottom line is why is this seagrass declining. And for anybody that has questions, there's a poster session with student Avantika Dutt, who's been indispensable, done great work, and I'll be on that session if there's any additional questions. And again, we're looking to expand the team. We're open to ideas. We are on an absolute mission, and there's the team. We're on an absolute mission to bring as many people in as possible, get as many ideas, because the seagrass is so critical to the aquatic life. We will not rest until we help identify what the problem is, so then we can solve it. Thank you.
Great. Thank you very much, Scott. Very, very interesting work going on there. And you did really well on time. You actually finished a little bit early. We are going to move on to Emily Grason, and also her, Dr. Emily Grason from Washington Sea Grant, wonderful institution. And also her co-authors are Adrianne Akmajian and Dawson Little from the Makah Tribe. And they are going to be talking about invasive green crabs on in Washington's National Park. And this is green crab are a pretty important invasive that have come into our area. And so this is quite a hot topic that I'm interested to hear a little bit more about. So without further ado, Emily, if you would like to take it away, I'd love to hear what you have to say.
Thanks, Stephen. I will be kicking us off today, and we'll be joined by Adrianne and Dawson, my colleagues who work on green crab with Makah. But you can reach out to any of us if you have questions about what we talk about today. So European green crab is a relatively recent arrival to Washington state, but it has a global history of damaging fisheries and ecosystems through predation and competition. It was first discovered in Washington in 1998 in coastal estuaries, but it didn't actually establish or grow populations until starting roughly a decade ago. The first discovery of any green crab across the Salish Sea portion of Washington's coastline didn't take place until 2016, and in fact occurred on San Juan Island. This picture in the bottom left showing your crab is showing that capture at a site that's very close to English Camp. The detection occurred as part of a 68-site statewide monitoring network. This is the program I work with at Washington Sea Grant that's called Crab Team. We started monitoring 6 sites in the San Juan Islands, 2 of which are on San Juan itself in 2016. I'm showing here the map on the top left has those 6 sites highlighted. These traps not only record the presence of green crabs, but also all other species in the traps. And that creates a really rich ecological record that helps us learn about these pocket estuaries and not just about green crabs, but potentially their impacts. On the south end of San Juan Island, the site in National Park Service property is Third Lagoon near American Camp. And no green crabs have ever been captured in the monitoring that's taken place there. So good news. Up on the north end of the island, there's a marsh at Westcott Bay, which is pretty close to English Camp. Since capturing that first crab in 2016, a total of 6 green crabs have been captured at the site. The most recent one hasn't been captured since 2019. So very, very low numbers. And that includes several large-scale follow-up trapping efforts to look outside of that one particular monitoring sites, including National Park Service property, Henry Island, Roche Harbor. Again, no findings of green crabs outside of this specific marsh. On the right here, I'm showing a map that more broadly across Salish Sea geography, green crabs have started to grow in populations, especially in the last 4 to 5 years. And the San Juan Islands so far remain relatively protected. However, we expect that this could change in terms of the habitat they use. And in particular, they're much more abundant in Washington's coastal estuaries than they are in these inland shorelines. And to talk a little bit more about the changes we've seen on the coast, I'm going to turn it over to Adrianne.
Thanks, Emily. So for those who aren't familiar, the Makah Reservation is located at the furthest northwest tip of the Olympic Peninsula, and it's adjacent to Olympic National Park shoreline as well. And we first found green crab on the Makah Reservation in 2017. We've since caught nearly 48,000 crabs on the reservation. You can see from the figure on the bottom left that catch total crabs varies between years. We had a large peak in 2022, a year with very high juvenile recruitment, but a lot of this variability has to do with changes also in trap types and areas that we were testing and setting traps. Over time, we've changed where and how we set traps. So when we first started trapping, you can see the figure, the map on the right. It was primarily in the Waiach and Skaggs rivers, which are estuary habitats. And we've since expanded to areas like the near shore of Nia Bay, as well as rocky intertidal areas at Waiach Point and Mad Bear Reef, which tend to be like a rocky intertidal bench, which is why we were interested in trapping near the Ozette Reservation, which Dawson.
Thank you, Adrianne. So this past summer, from July 5th through 7th, myself, along with other tribal staff, partners, and volunteers, hiked to Cape Alava with the goal of prospecting for invasive green crab. We deployed a total of 12 half-inch mesh shrimp traps over one night near the border of Olympic National Park and the Ozette Reservation. During this effort, we caught a total of 14 invasive green crab for a catch per unit effort of 117 crabs per 100 traps. This is a similar and in some cases higher catch rate compared to areas we trap more regularly farther north on the reservation. And these crabs were in the 60 to 80 millimeter range, suggesting they've been there for around 2 to 3 years. We believe this detection warrants a follow-up to attempt to further assess the extent of invasive green crab in this area. So in 2025, our team is currently planning a follow-up focusing more on areas located on the actual Ozette Reservation, with hopes of collaborating with other groups located in the region.
And with that, we'll just leave you with a slide on how you can help.
Fantastic, really timely stuff, and really interesting work. So thank you very much for this collaborative effort with Washington Sea Grant and the Makah Tribe. Moving on, and once again, questions in the questions and answer. Everyone will be around to respond at the tail end of this session. Our next talk is going to be from Andy Marquez from Stanford University, who's been doing some work with us at Olympic National on with some of our long-term monitoring of sandy beach critters that live inside. So charismatic millie to microfauna. Andy, you can take it away now.
Okay, thank you, Steve. Can people hear me and see my slides?
Yes.
Cool. Great. So I'm going to be talking about some work that I've been doing as an intern with Olympic National Park. I'm going to be kind of describing some of the sand beach monitoring that happens here at Olympic, as well as the work that we've done to try to identify environmental drivers of sand beach community structures at Olympic National Park. So if you're lucky enough to visit a sand beach at Olympic National Park, you might be met with a landscape that looks something like this. And at first, it might not look like too many organisms are living here, but if we look just beneath the surface of the sand, we find actually really abundant and biodiverse communities of marine invertebrates that are living here. These marine invertebrate communities consist of organisms like isopods and amphipods and a wide variety of worms. Sand beach ecosystems are really important for Olympic National Park because they make up 30% of Olympic National Park's intertidal zone. They also provide essential ecosystem services without which the greater coastal communities here at Olympic could not function. Some physical ecosystem services they provide are storm buffering and water purification, and the organisms that live in sand beach ecosystems drive nutrient cycling along our coast, as well as provide essential food sources for other organisms living along our coast. Speaking to the importance of sand beaches, the North Coast and Cascades Inventory and Monitoring Network has been conducting sand beach monitoring at Olympic National Park since 2005. These are some figures to help visualize the sand beach protocols here at Olympic National Park. There are 6 sand beaches that are visited annually since 2005. Shy, Sand Point, Cedar Creek, Second Beach, Ruby Beach, and Claylock Beach. Every year at each of these beaches, we set up 3 transects that might look something like the figure on the right here. The first part of our monitoring protocol is we walk along the center of the transect along these blue dots with a stadia rod, and we measure the beach elevation, and this allows us to measure the slope of the beach that we're looking at. Every 7.5 meters, we take 4 sand cores, and we sift through the sand and physically count every single marine invertebrate that we encounter. And then every 15 meters to the left of the transect, we take a sand sample. And then we hike the sand samples out, and we essentially process the sand in our lab, and we can look at the various sand grain sizes for the sand at each beach. So given the sand beach monitoring protocols that happen here at Olympic National Park, we have a really rich data set of beach profiles and beach slope, sediment compositions, and infaunal community structures for the 6 beaches that we monitor at Olympic National Park. And given this rich data set, the research question that we wanted to answer is, how does the abiotic environment influence the communities living in the sand beaches of Olympic National Park? And this is a really important research question, because if we hope to protect these communities in the face of a changing climate, then we need to understand which environmental variables are most important for structuring the communities here at Olympic National Park. So the first step to answering our research question is to identify differences in the infaunal communities among the sand beaches that we study or that we monitor. In order to do this, we used a non-metric multidimensional scaling analysis. We can see this here on the left. And this is essentially a way to visualize differences among the beaches that we monitor. So points that are closer together are more similar, and points that are further away are more dissimilar. And the multiple points for each beach represent a different sampling year. And then here on the right, we can see an anosim, or an analysis of similarity. And this is essentially a way to quantify differences in infaunal communities for the various beaches that we monitor. And we can see that there are various amounts of differences in the infaunal communities here at Olympic National Park that are all statistically significant. So the next part to answering our research question is identifying the environmental variables that drive these differences among the sandy beaches that we monitor. And when we do this, we found that mean beach slope was a really important environmental variable that drives these differences in infaunal community structures, followed closely by fine sediment, or mud. And next steps for our work include including environmental variables like sea surface temperature that might also be contributing to differences in the infaunal community structures of the sand beaches that we monitor. Special shout out to Bill Bacchus and Steve Radkin that lead the sand beach monitoring here at Olympic, the 2024 intertidal monitoring crew, and all of the intertidal monitoring crews, past, present, and future.
Awesome. Nice job, Andy. Fantastic work. What else am I going to say? But no, really, really good. We're going to now move. That was a great bridge from biological and physical to moving over to some physical considerations now. So Taylor Skaggs from the Washington Department of Ecology is going to talk to us about cobble berm monitoring. So once again, sort of sediment type things, looking at nature-based coastal protection structures on the Olympic coast. So Taylor, take it away.
Thank you so much for the introduction. All right. Can everyone see my screen okay? All right.
Yes.
Hi, my name is Taylor Skaggs, and I will be presenting on behalf of the Washington Department of Ecology's Applied Coastal Research and Engineering section. This presentation will be covering our ongoing monitoring of a cobble berm in Olympic National Park to inform designs of nature-based coastal protection structures. The primary reason that we are monitoring the shoreline, including the cobble berm that falls within South Beach Campground in the southern extent of Olympic National Park, is to provide an analog to dynamic revetments in other areas of the state. This comparison is intended to inform the design and adaptive management process for dynamic revetment projects. The stretch of shoreline consists of a sandy foreshore and a cobble berm higher on the beach and can also be referred to as a composite beach since it is made of both sand and gravel or cobble. Cobble berms are more resilient to high waves and water levels in comparison to pure sand beaches, as the cobbles on the upper beach absorb wave energy and can move with the current. Inspired by the resiliency of natural composite beaches, engineers have designed shoreline stabilization structures called dynamic revetments, such as the North Cove dynamic revetment, which can be seen on the right. Dynamic revetments are intended to function as a nature-based approach to shoreline protection from upland erosion for high-energy environments and are more adapted to changing conditions such as sea level rise. These diverge from hard shoreline armoring, which consist of static structures with fixed location, geometry, and larger materials such as riprap or concrete. However, dynamic revetments are a relatively new solution compared to the more conventional measures, and there's no published engineering guidance for dynamic revetments at this time. This can lead to challenges during design and management, and why monitoring both natural and artificial structures to understand their behavior is so important. As part of the ongoing monitoring effort at South Beach, we complete end-of-season surveys, including topographic monitoring of cross-shore profiles, walking the tow berm, and collecting photos and sediment samples. We also have an annual surveying event in which rocks that have been tagged with RFID trackers are located and logged for their location on the beach. My colleague Heather presented in detail on all of these methods at last year's North Coast and Cascades Science Days, and there were a couple requests for more information on our rock tracking work, so I will be focusing on that data today. The addition of RFID trackers is part of the effort to more accurately predict cobble movement as part of dynamic revetment design, since the angle of wave incidence and geometry of a berm heavily impact cobble movements. To employ this process, we took 150 cobbles from South Beach, brought them back to our lab, weighed, took dimensions, and drilled holes into the cobbles and installed RFID tags, which you can see the little cap in this photo of cobble 5332. The cobbles were then installed during the summer 2019 survey at various elevations along 5 transects. During the annual surveys, these cobbles are tracked and then logged for their new location. After logging where the cobbles were found during our annual tracking, we calculate the direction and distance that they move both from their original location and their last known location. This plot displays the movement of 3 of the cobbles over the 5-year span, with the gray X's showing the original placement of all 150 cobbles, and the colorful ones showing the start points for the only 3 that we have located during all 6 of our tracking surveys. Over this 5-year span, we have seen movement that is consistent with the seasonal transport cycles, as most of our surveys have occurred during the winter when there is northward transport. In addition to the alongshore transport, there is also both landward and seaward cobble movement, which exemplifies the upslope cobble transport mechanism, allowing cobble berms to be resilient to high energy wave climates. This method is not perfect for tracking, as we have experienced a declining annual recovery rate. The main cause is not that they have been pushed out of our search area, but that the RFID tags can only be located down to 1 meter of depth, which is significantly less than the overall depth of the berm. While we are only capturing these data points on an annual basis, it is very clear that the cobble berm at South Beach is incredibly dynamic. Over the past 5 years that we have been surveying, its overall geometry has remained stable, exemplifying its resiliency in a high energy wave environment. Composite beaches like South Beach are currently underrepresented in scientific literature compared to sandy beaches, and there is still so much for us to learn about how cobble berms change. Our overall goal is to improve engineering tools guiding the planning and design for future dynamic revetments. One of the ways that we are trying to apply this research is by comparing our data from South Beach to empirical equations for sediment transport. The berm at South Beach has proven to provide natural shoreline protection despite significant observed mobility of cobbles, highlighting the need for continuous monitoring and management of dynamic revetment structures following their construction. With sea level rise and eroding coastlines, it is now more important than ever for us to be researching nature-based erosion preventative structures like dynamic revetments and to use natural cobble berms like the one found at South Beach as an analog to help us succeed. Thank you so much for listening, and thank you so much for all of the Acre team. If you are interested in learning more or coordinating with Acre, please reach out to us at this email. Thank you.
Fantastic. Thanks a lot, Taylor. Really cool stuff. Anyone who's ever gone, particularly in the Clay Lock area, and seen these cobble berms sitting on top of sand beaches has probably always wondered, like, how are these maintained? And that you've gone a long way to addressing some of the curiosity on that. So love the work. Love to see it continuing going on. I will add that once again, questions in the Q&A. I noticed some people have been putting questions in the chat. That's okay also. I'll be visiting both of those at the end, but if you can try to get in the Q&A, that would be great. All right, the next talk also is sedimentary in nature and delves a little bit back into the biology of bull kelp, an iconic kelp in the Pacific Northwest. And so Spencer Hill from the University of Washington will be talking about sediment and sediment densities affecting bull kelp life histories. Okay, Spencer, let's hear it.
All right. Hello, my name is Spencer, and last fall I had the opportunity to study at UW's Friday Harbor Laboratories up in San Juan Island. And as a part of that, I got to look at how sediment densities affect early stage bull kelp or Nereocystis Luetkeana stages in sandy environments. So for an overview of bull kelp, its range is from Southern California to Southwest Alaska. It serves a variety of ecological functions. It is a carbon sink, pulling carbon from the water, which in turn pulls CO2 from the atmosphere. It is a physical barrier from waves, which protects shores from erosion and intertidal communities. And it provides structure and major food source for organisms in kelp forests. Unfortunately, bull kelp is declining in many regions due to a combination of local and global stressors. In South Puget Sound, as one example, bull kelp has declined by 63% over the last 150 years. So it is a quite substantial issue we're facing. So for the life cycle, it is annual. So it reproduces and dies off every year. In some cases, it can live up to 18 months, but that's pretty rare. It has a multi-stage reproduction. So the large bull kelp that most people are familiar with is known as the adult sporophyte stage. This will release spores that develop into male and female gametophytes. The male gametophytes will release gametes, and once the female gametophytes become fertilized, they will develop an embryo, which develops into the juvenile sporophyte that then grows back up into that large adult sporophyte stage. And when they're in the sporophyte stage, they can grow 15 to 25 centimeters a day, which is how they're able to achieve such great lengths in only a year's time. We focused on specifically the microscopic development stages from spores to juvenile sporophyte. So sediment could be a potential stressor to these organisms because sporophytes need to anchor themselves to substrate to withstand wave forces. But large sediment disturbances like storms can alter sediment dynamics. This can lead to erosion and burial of these benthic communities, including kelp. And with increased urbanization and dam removals, there's going to be more terrestrial sediment entering marine environments each year, posing a larger and larger risk. And for why it could be bad, it could also restrict access to nutrients or light, which is very important, especially at these microscopic stages. So we collected reproductive patches known as sori in San Juan Channel and settled spores onto glass slides in lab. We would then put these slides into beakers and then add a solution of sterile seawater, nutrients, and a sediment concentration, with the sediment then resting and settling onto the glass slides. We would put these in the refrigerator to mimic seawater temperatures naturally, and they would develop. And then each week, we would take these slides out of the beakers and put them into Petri dishes, and then we would photograph the slides with a microscope camera. We had 4 sediment treatments, a control with no sediment, and then 3 different levels trying to mimic possible sediment concentrations found throughout the San Juan region, and 2 light levels, dim and bright, because we wanted to see if the main issue was sediment restricting light access to kelp. So for our photo analysis, we came across some issues. Originally, we wanted to just count survival of kelp gametophytes over time and produce quantitative data that way. But as kelp developed, it would grow larger and kind of grab onto others. So it became difficult to distinguish individuals photo, we weren't sure if this was one kelp or if this was a dozen or more. In some cases, specifically controls and low sediment densities, there was just too much kelp surviving, and it became way too dense and layered to be able to count individuals. And then in higher sediment densities, we found an interesting issue with kelp holding onto sediment almost like little hats. And it became very difficult to see what was going on under those little sediment pieces and count them that way. We were, however, able to get some qualitative observations with gametophytes showing to be resilient. They were growing even in the high sediment treatments and developing, although definitely at a slower pace. The sediment did have an effect, with more sediment in general equating to less growth, survival, and development of gametophytes. And interestingly enough, light did not appear to affect gametophyte success, so it may be more of a nutrient issue with sediment. So future research could look at different methods to remove sediment to be able to produce that quantitative data. We could study the mechanisms that allow bulk kelp gametophytes to stick to sediment, the effect of different sediment sizes, as we mainly focused on fine sediment, and the effects of suspended sediment and flowing water, as opposed to stagnant water and settling sediment. I want to thank Rebecca Hansen, the lead researcher, Serena Maiwal, my fellow lab technician, and UW Friday Harbor Laboratories for giving me this opportunity. Thank you.
Fantastic. Really cool stuff, Spencer. So thank you very much. Sediment on kelp. You know, our region is an incredibly biodiverse region. The Olympic Coast is arguably one of the most biodiverse intertidal zones in the lower 48. And so we're going to now move to our next talk to concentrate on kelp and rockweeds and their diversity. So Dr. Christopher Janusik from Oregon State University, not the University of Washington, as is in the abstract from Oregon State. So sorry about that, Chris. And well, I'm really looking forward to hearing about your diversity talk. So take it away.
Yeah, thank you. Good to be with you this afternoon. So I am faculty at Oregon State University, and I also teach occasionally at Cal State Long Beach, but this is actually an independent side project that I've been working on for years. It kind of grew out of my interest in tide pooling and visiting sites up and down the coast. So it's taken a while to pull this together, but this has been a fun project to look at the distribution of kelps and rockweeds along our Pacific coast. So these large brown seaweeds have key trophic and structural roles on rocky shorelines. They're foundation species that provide habitat for other organisms, a lot of productivity, but they're also threatened currently by climate change and impacts like urchin overgrazing. So this figure below, taken from a paper a couple of years ago, shows the decline in kelp at a site in Northern California, Mendocino County. So urchin grazing and these impacts are threatening these communities along the coast. There were two objectives in this project. The first was very biogeographic in nature. It was to determine the geographic patterns of individual species as well as biodiversity across the whole California current ecosystem, so Washington, Oregon, and California, and then also to establish a regional data set that could be valuable for studying long-term change in these communities. So the method I used was fairly simple in a lot of ways. It consisted of rapid surveys of individual sites. These sites were 200 meters long. I studied about 100 sites along the coast from Southern California up to Olympic National Park. There were 3 Olympic sites in this particular study. A couple of other national parks were involved as well. So Redwood National Park, Golden Gate, and Point Reyes in California. And the method consisted of going out to the intertidal for about 3 to 5 hours, surveying the intertidal and the shallow subtidal at low tide, crisscrossing the site. So this little diagram here is just an exercise app that shows you my path up and down the site multiple times to assess the community structure. And at each site, I measured species presence or absence, their abundance on kind of a log scale, so sort of a crude quantitative measure of abundance, and then species diversity. All right, to jump into just a couple of slides of the results, we found 29 species across this region. 2 of those species were non-native, and those were both fucales or rockweeds. And the species could sort of be divided into 3 groups. First, we have an example here. This is feather boa kelp. These are species that were really common. So they were present at anywhere from 30 to 50% of the sites, are kind of ubiquitous across the Northeast Pacific. The second group are subdominants or occasional species. So these were present at maybe anywhere from 10 to 30% of the sites. And then finally, we had a group of species that were quite rare, or they may have been abundant, but only in local geographic regions. One of the things I wanted to do was look at the species composition up and down the coast. And so I used NMDS, and a couple of talks ago, you saw an example of that from sandy beaches. So again, you can interpret these points by looking at how close they are together, and pairs of points that are close together have similar species composition. So one of the things that fell out in this analysis is that there's a pretty strong biogeographic gradient, which isn't all that unexpected because we have gradients in upwelling and temperature and tide range moving from Southern California up to Washington. The three blue dots here, kind of at the middle right of the graph, these are the three Olympic National Park sites. And they do cluster, interestingly enough, with a lot of the sites in Northern California. One other thing we've taken a look at so far with the data is the geographic patterns of species diversity. So on the left are box plots of rockweeds. These are more in the mid-intertidal, sometimes in the high intertidal. And on the right are the kelps. These are more in the low intertidal or the shallow subtidal. And these show opposite patterns of species diversity with geography. So the rockweeds are most diverse actually in Central California. There's a couple of endemic or rare species there that boost their diversity. And then they're only about half as diverse from Northern California up to the Washington coast. Kelps kind of show the opposite pattern. Kelp diversity is lowest in Southern California, where there's just typically a few species in the intertidal, but then that diversity grows up into the Pacific Northwest. So just a concluding slide here, the California current has distinct assemblages of large brown seaweeds, rockweeds, and kelps. The Southern and Central California assemblages are particularly unique, which is interesting. These two groups of foundation species show different latitudinal patterns up and down the coast. And by doing these kind of rapid but numerous studies, we're able to find the distribution of short-range endemics as well as rare species that you might not see at your typical site. And these species are important because they increase diversity at a regional scale. They also might be important species that we want to track over time because they could be climate change indicators if they're dropping out or just can't be found anymore. So the next steps with this project, again, it's a side project, so this is taking me forever, but I'd like to look at the local and regional scale drivers that affect these communities, things like slope, topography, and upwelling. And I'd like to thank the National Park Service, again, was able to work at 4 sites, which was really cool, as well as the UC Natural Reserve System in California. Thanks.
Fantastic. Really, really cool stuff, Chris. Thanks a lot. And I always love side projects that are productive. So really cool. So thank you all to the presenters because I thought that was a great session. Cut a very wide swath through marine themes. And so I think at this point now, maybe what we'll do is we'll move into, while we've got several minutes left, we'll move into answering some of the questions from the Q&A and in the chat. I know that some folks have already addressed some of those questions, but I'll sort of go through some of these a little bit. Let's see. John Betch asked whether might NDMS and variable selection eventually lead to being able to model species groups and community composition shifts with changes in abiotic conditions. Is there enough data for that? I'm curious to hear what you might have to say to that, Andy.
Totally. I think this is a really great question. And I think once we identify environmental variables that are really important for sand beach community structures, I think that a logical next step would be to use modeling to maybe predict changes in community structures, given which different environmental variables are important. I don't know that I would have time to do that as a part of my internship, but I bet Steve would definitely be interested in like spearheading that even after my internship is over. But I think there's definitely plenty of data there to use modeling, to look into changes in community structures.
Yes, but of course. And Andy, I think there was also another, we're on you. I think there was another question regarding, let's see, where was it? I thought it had to do with how much work you were doing on this, but at any rate, how many years has Jennifer Hagan from Quileute asked how many years has this work been occurring, and were you able to see effects from storms that move sediments?
That's a really great question. Sand beach monitoring at Olympic National Park has been happening since 2005, I believe. Steve, you should correct me if I'm wrong. And so now that we know that beach slope is an important environmental variable for looking at sand beach community structures, I think it would be a really good idea to go back and look at years where there were significant changes in beach slope and see if like there were significant changes in the infaunal communities as well. So thanks for that question.
Excellent. Bill Bacchus had a question for you, Chris. Any idea why there was a single outlier in the Washington samples on the NMDS plot?
Yeah, that's a great question. That particular site, I believe, was near Anacortes. So that was in the Salish Sea. And I think there are pretty large differences in communities between the Salish Sea and the outer coast of Washington in terms of wave exposure and probably upwelling and other processes. Again, because this was kind of an opportunistic study, I haven't been able to do as many sites in Washington. I don't live in Washington, so it's been a little bit tough to get up there, but I would love to survey more sites in Puget Sound.
Great. And I have a quick question for you, Chris, which is sort of related to these latitudinal trends that you saw for the Fucales and then also for the kelps, which were sort of in contrast to each other. How do those compare to, and you may not be able to answer this, but how do those compare to other taxonomic groups of algae, like red algae and things like that? Are there similar sorts of trends that mirror those or that are in contrast to those?
Yeah, that's fascinating. Red algae are really very diverse in the California current ecosystem, but their composition does change a lot as you move from kind of the warmer waters of Southern California up to, much cooler and more nutrient-rich waters of Oregon and Washington. I don't know that anyone's looked at that, and I think that would be a really cool thing to look at. I work a lot actually on salt marsh plants, so I have some similar data up and down the coast in salt marshes, and there are some interesting latitudinal gradients as well. And salt marsh plant diversity seems to peak kind of around Southern Oregon, Northern California, which is kind of fascinating. It drops off in further south in California, and it's not quite as diverse in Washington. So I think there's some kind of just fun biogeographic questions with these different kinds of groups up and down the coast.
Great. Awesome. And we might be running out of time. I'm going to defer to you, Hazel, on that, but I've got a lightning round of questions of my own. Let's see. One of these was for, oh, good God. Taylor, with regards to the cobbles that you attach these RFID tags, did you find in terms of being able to recapture those things, did actually drilling holes in those actually damage them? And across years, actually, did you lose them because some of them broke open and you didn't get the RFIDs back?
So our likely theory for it is that because the cobble berm changes so much during the seasonal changes that we have, or the seasonal cycles that we see, that it's most likely that they're buried underneath other rocks. And once they get down to 1 meter below the surface, we can't reach them anymore. So they won't show up if we try to track them, because it's basically like a metal detector. The holes don't really weaken them that much, but they were very time consuming to drill.
Gotcha. Okay, cool. Just a little question that I had. And then also one quick question for you, Scott, on the chemistry. You put these pom-poms out that you detected a lot of diesel and some metals. What was the relationship, or have other people actually looked at the relationship in terms of diesel contaminants and actually affecting, in terms of toxicological studies, actually affecting eelgrass, that link there?
Yeah, that's a great question. We have an expert team, and there's no question that, diesel does affect negatively, seagrass. And the old adage is true: 1 gallon of oil contaminates 1 million gallons of water. It spreads pretty quickly. And then the issues on the metals is on the nutrients. So do we potentially have a combination of the PAHs affecting the seagrass with the manganese or the phosphorus, potentially sparking some algal bloom we haven't identified, or or bacteria we don't know?
Certainly a lot going on in the marine environment. It's a very complicated system, biologically, microbiology, and chemically, and all those things are related. I see by the clock on the wall that it is 2:02, and so Hazel, am I right that our session is over, or can I keep going
on? But take these conversations over to the poster session. Scott mentioned that he's going to continue sharing about his work with his colleague there. I invite you to just jump on over into that main room and continue chatting at will, including folks who are not on the screen right now.